We often imagine DNA as a static blueprint of life. It's a molecule with a stable structure passed down through generations. We tend to think that it remains largely unchanged, but this perception couldn't be further from the truth. The DNA inside our cells is under constant attack. From the ultraviolet radiation of the sun to various chemicals in our environment, our DNA is constantly bombarded by agents that can damage its delicate structure. Imagine a bustling city with towering skyscrapers. These skyscrapers represent our DNA molecules. Now, picture these buildings under constant bombardment from wind, rain, and even the occasional earthquake. This is the reality of our DNA. But just like a city has its repair crews, our cells have evolved sophisticated mechanisms to repair this damage. These repair mechanisms are essential for maintaining the integrity of our genetic information. Without them, our cells would accumulate errors in their DNA. These errors could lead to cell death or, even worse, cancer. For decades, scientists believed that cells had no way to fix damaged DNA. This belief stemmed from the observation that mutations, changes in the DNA sequence, seemed to be permanent. Mutations were seen as the driving force of evolution. They provided the raw material for natural selection to act upon. However, this view began to change in the mid-20th century. Researchers began to notice that cells could sometimes recover from what seemed like lethal doses of radiation. It was as if the cells had found a way to undo the damage. This observation led to the startling realization that cells must possess some intrinsic ability to repair their DNA. The discovery of DNA repair mechanisms revolutionized our understanding of genetics. It revealed that DNA is not a static molecule but a dynamic entity capable of self-healing. One of the earliest clues about DNA repair came from studies on bacteria. Scientists found that bacteria exposed to DNA-damaging agents like UV light were more likely to survive if they were allowed to rest in a nutrient-rich medium afterward. This period of rest, it seemed, gave the bacteria time to repair their damaged DNA before resuming normal growth and division. Think of it like taking your car to the mechanic. If your car has a flat tire, you can't just keep driving. You need to stop, jack up the car, remove the damaged tire, and put on a spare. Similarly, bacteria need time to stop their normal cellular processes and focus on repairing their damaged DNA. This finding suggested that DNA repair wasn't just some passive process. Instead, it was an active, energy-demanding process that required the cell to dedicate resources to fixing its genetic blueprint. This discovery hinted at the existence of a dedicated DNA repair machinery within the cell, poised to spring into action when needed. A pivotal experiment in the 1950s by Jean Weigle provided further evidence of DNA repair. Weigle was studying a virus called Lambda phage, which infects bacteria. He exposed Lambda phages to high doses of UV radiation, enough to severely damage their DNA. He then used these damaged phages to infect bacteria. To his surprise, Weigle found that some of the bacteria still produced new phages, even though the phage DNA was heavily damaged. This indicated that the bacteria were somehow able to repair the damaged phage DNA, allowing the phages to replicate. Weigle's experiment was a breakthrough. It showed that DNA repair wasn't just a phenomenon limited to the cell's own DNA. Cells could even repair the DNA of invading viruses. This finding highlighted the universality and critical importance of DNA repair across different life forms. Section 5. Deciphering the SOS response in bacteria. Weigle's work, along with other studies, paved the way for the discovery of the SOS response. The SOS response is a global response to DNA damage in bacteria. It's like a cellular alarm system that gets triggered when the cell senses significant damage to its DNA. This response is not limited to a single repair pathway. It involves a coordinated activation of a whole suite of genes involved in various aspects of DNA repair, DNA replication, and cell cycle control. Imagine a city suddenly hit by a massive earthquake. Buildings are damaged, roads are blocked, and communication lines are down. In response, the city activates its emergency response system. Firefighters rush to extinguish fires, rescue teams search for survivors, and engineers work to restore essential services. The SOS response is the bacterial equivalent of this citywide emergency response. When the SOS alarm is sounded, the cell channels its resources towards repairing the damage, preventing further damage from occurring, and ensuring the survival of the cell. Section 6. The two paths of DNA repair, accuracy versus speed. The SOS response encompasses two main types of DNA repair mechanisms, error-free and error-prone. Error-free repair, as the name suggests, aims to restore the original DNA sequence with high fidelity. It's like having a skilled craftsman meticulously repair a broken vase, ensuring that every piece is put back in its exact place. Error-prone repair, on the other hand, is a last-ditch effort employed when the DNA damage is so extensive that the cell has no other choice but to risk introducing mutations. It's like having a hurried repair job done on the vase, where some pieces might be glued back incorrectly or even replaced with mismatched parts. While error-prone repair can introduce mutations, it also buys the cell some time to survive the immediate crisis. Some of these mutations might even prove beneficial in the long run, providing the cell with a selective advantage in a changing environment. Section 7, RecA, the guardian of bacterial genomes. At the heart of the SOS response lies a key protein called RecA. RecA is like the general of the bacterial army, coordinating the troops in the face of DNA damage. When DNA is damaged, especially by UV radiation, it creates single-stranded DNA, which acts as a signal for RecA. RecA binds to these single-stranded DNA stretches, forming long filaments. These filaments act as a platform facilitating the activation of various SOS genes, including those involved in both error-free and error-prone repair. RecA is not just a passive observer. It actively participates in the repair process. It helps mediate homologous recombination, a process where a similar DNA sequence from another chromosome is used as a template to repair the damaged DNA. This ensures that the repair is as accurate as possible. Section 8, from bacterial SOS to human health, a universe of DNA repair. The study of DNA repair in bacteria has far-reaching implications, extending far beyond the microscopic world of these single-celled organisms. The principles discovered in bacterial DNA repair are highly conserved across different life forms, including humans. Many of the DNA repair pathways found in bacteria have their counterparts in eukaryotic cells, highlighting the fundamental importance of maintaining genetic integrity across all life. Understanding how these repair mechanisms work and what happens when they go awry is crucial for understanding the development of cancer and other diseases. The SOS response in bacteria serves as a powerful reminder of the dynamic nature of DNA. Our DNA is not an inert blueprint, but a dynamic molecule, constantly under attack and constantly being repaired. As we delve deeper into the molecular mechanisms of DNA repair, we gain a deeper appreciation for the intricate dance between our genes and the environment. We begin to unravel the secrets of how life maintains its order in the face of constant change, a testament to the enduring power of evolution.